Hi, I'm Adam Byron, and welcome to this brief presentation summarising our article about cell adhesion proteins and how they regulate cell behaviour. Cell adhesion is essential for multicellular life to exist. Cell adhesion is mediated by adhesion receptors, such as integrins, which are proteins on the surface of cells. Adhesion receptors attach to the extracellular matrix, a complex mixture of proteins and sugars that surround cells, providing mechanical support and biochemical signals. During cell adhesion, adhesion proteins inside cells are recruited to adhesion receptors to form multi-protein assemblies called adhesion complexes. The networks of adhesion proteins and their interactions help cells sense their surroundings, and this determines whether cells move, divide, or die. When cell adhesion goes wrong, these processes malfunction, and disease often results. So working out how cell adhesion systems function as interacting networks is currently under intense scrutiny, and this is the focus of our article. But adhesion proteins do not just spend their time near the cell membrane in adhesion complexes. Recent studies have uncovered wider roles for adhesion proteins away from adhesion receptors, and the second half of our article highlights the important functions of adhesion proteins in diverse subcellular locations. The proteins in adhesion complexes connect integrins at the cell membrane to the cell's cytoskeleton, and they have been collectively termed the adhesome. Proteins in adhesion complexes can now be isolated biochemically from cells. We can identify and quantify the proteins using proteomics, and we can analyze the data computationally using bioinformatics. The computational integration of multiple sets of adhesion proteins has now enabled the definition of a meta-adhesome, incorporating data from different types of cells. From this analysis, outlined in this slide, a core set of 60 frequently identified proteins was identified, and this was termed a consensus adhesome. The assembly and disassembly of adhesion complexes is tightly regulated to enable cells to attach to and detach from their surroundings, to migrate effectively, and to control other cellular processes. But precisely how this happens is poorly understood. As outlined in this slide, there appear to be roles for certain adhesion proteins at specific stages of adhesion complex formation. The levels of adhesion proteins during adhesion complex assembly and disassembly have been measured using proteomics. The measurements revealed distinct patterns of protein recruitment shown in this slide. A modular or hierarchical system of adhesion complex assembly and disassembly might allow the rapid fine-tuning of adhesion signaling in cells. But despite their name tag, adhesion proteins are not just found associated with cell matrix contacts. For example, the adhesion protein focal adhesion kinase, or FAK for short, can be found in the nucleus of normal cells when they've undergone cellular stress. It turns out FAK is also present in the nucleus of skin cancer cells, where it profoundly influences the expression of genes. Using a proteomic, bioinformatic, and network approach outlined in this slide, it was discovered that FAK binds to proteins in the nucleus that control gene expression. These proteins regulate the expression of chemokine genes, for example, which encode signaling proteins that are released by cells into their surroundings. The detailed mechanisms by which FAK regulates gene expression in a selective way are still to be worked out, but it is clear that FAK's control of gene expression leads to the release of chemokines that attract regulatory T cells from the immune system. In skin cancer, this results in the cancer cells evading destruction by the immune system, which leads to tumor growth. Importantly, there is no detectable nuclear FAK in normal skin cells, suggesting that nuclear accumulation of FAK is linked to the pathology of skin cancer. This then raises the possibility that nuclear FAK could be targeted therapeutically to treat cancer. Only by understanding the networks of adhesion proteins and their interactions will we find out how cell adhesion signaling coordinates so many aspects of cell biology. We are funded by Cancer Research UK. Thank you for watching.